In this video, we are going to talk about blood agar. Blood agar is also known as tryptic soy agar that is supplemented with 5% whole sheep's blood. And blood agar is an enriched media. It is just like nutrient agar. Tryptic soy is a general purpose supportive medium that has lots of different nutrients in there that most things can grow on, but it's enriched with that 5% sheep's blood in order to make it a better environment for really fastidious organisms like the streps. The streps grow really well on blood agar. So blood agar is an opaque red plate. It is very red and you cannot see through it under a normal basis. Blood agar is only differential. It's not selective at all. Any bacteria can grow on blood agar, but the way that they grow on blood agar tells you something about the bacteria. Specifically, it tells you if they are able to lyse red blood cells, something called hemolysis. There are 3 different types of hemolysis: alpha hemolysis, beta hemolysis, and gamma hemolysis. The first one I'm going to talk about is actually beta hemolysis. Beta hemolysis is complete hemolysis. So bacteria that display beta hemolysis produce enzymes and proteins that they can use to lyse red blood cells. And they lyse all red blood cells that they come in contact with, and they lyse them completely. You can tell if an organism is beta hemolytic because they will actually cause clearing on the agar. Here you can see everywhere that the bacteria are growing, you can actually now see through the agar in those areas. I like to take a blood agar plate and even put it against something with writing on it, and you can actually read through the cleared areas of blood agar. So if you see blood agar with clearing, this shows you complete hemolysis. Alpha hemolysis is incomplete hemolysis. These bacteria are able to lyse red blood cells, but they can't lyse them with the same efficiency as those that can lyse completely. So instead of seeing clearing on the media, instead the media turns somewhat green where the bacteria are. It might be a little bit hard to see in this video, but I know I have better pictures in the lecture or in the lab PowerPoint, and you can find lots of pictures of this online. So you can see everywhere the bacteria are growing, the media has actually turned green behind them. And the third type of hemolysis is gamma hemolysis. And gamma hemolysis is actually no hemolysis. Organisms that have gamma hemolysis are not able to lyse red blood cells at all. And so with gamma hemolysis, when you look at the blood agar, you can see the bacteria growing on the agar, but there is no color change to the media, there is no clearing. You can just see the colonies on the agar themselves. And every type of organism that we work with this semester will display some type of hemolysis. And in some cases, the kind of hemolysis that you see is very important in the identification of that organism.